Welcome to another screencast. This one is on glycolysis, the details of it. So we're going to first make sure that you have a working understanding here of the three phases, locations of aerobic respiration, the overall purpose of respiration, and then what redox reactions are. We're going to focus here on describing the reactants, products, locations, and purpose of glycolysis. So again, reactants, products, location, and purpose of glycolysis. The two subphases of glycolysis, as well as the purpose of NADH here. And ultimately, we want to try to get to this substrate level phosphorylation explanation, but this is that'll just be icing on the cake if we can get to that. So, big picture is aerobic respiration, and we are going to be discussing this part of the diagram here today, right, in this screencast. So we're going to be looking at glycolysis and what's going on in glycolysis. This diagram shows you the whole overall thing here. So in glycolysis, glucose is going to be oxidized during glycolysis, releasing free energy. And then ADP is phosphorylated, and NAD plus is reduced. So we're going to essentially oxidize glucose, and we're going to reduce NAD plus in its simplest terms. Right? Along the way, we're also going to phosphorylate ADP, right, to generate ATP. So this is the simplified way. And again, there's no other simpler way to explain it than with this. That said, oxidation reduction is a complex thing. So here we go. We're going to start with the glucose molecule, which is, of course, our organic molecule. So it's carbon, hydrogen, and oxygen. And in the first part of glycolysis, there's this energy investment phase. So this is one of the subphases, the energy investment phase. So this glucose molecule is relatively energetically stable. We need to use two ATP molecules here to energize it a little bit. So this seems odd, I know, because right now we're going down to ATP, right? And we're trying to make ATP over here as an end product. We first need to input a little bit of energy into this system here. So we're going to use these two ATP. This glucose becomes now more activated. But then in the second phase here, the energy payoff phase, the glucose is going to continue to be oxidized, and we're going to, in the process, phosphorylate ADP to generate ATP, and then we're going to do a reduction here. So this is, we're going to reduce NAD plus to NADH. The end product then are these pyruvate molecules down here. And you notice that we're having two pyruvate molecules. And these pyruvate molecules are organic molecules as well. So they have carbon, hydrogen, oxygen. Now the difference between the glucose, carbon, hydrogen, oxygen and the pyruvate carbon, hydrogen, oxygen is the way that they are arranged, the number of carbons and hydrogens and oxygens there. So this glucose up here is a 6-carbon molecule, and these two pyruvates down here are 3-carbon molecules. So we have two 3-carbon molecules there. Okay. So what happens is, again, in glycolysis, is we're going to take this 6-carbon glucose molecule and we're going to split it. Right? This is where we get the lysis from. Right? Split or divide glucose, which is one 6-carbon molecule, into two 3-carbon molecules. So we're going to go a 6-carbon, and it's going to be split into two 3-carbon molecules. This splitting here frees up some energy to do this phosphorylation and frees up some energy here to do this reduction. So over here, we're going to oxidize glucose to pyruvate. This is how we say this. We're going to oxidize glucose to pyruvate, and we're going to reduce NAD plus to NADH. these redox reactions overall net are negative delta G net. We're going to use this negative delta G to then drive the positive delta G phosphorylation of ADP to ATP. So that is seen down here in this box, right? Where when I oxidize glucose to the 2 pyruvates, right, this is a negative delta G. This phosphorylation of ADP to ATP is a positive delta G. This reduction of NAD plus to NADH is a positive delta G. So this negative delta G right here is more than this plus this. Looking at these animations there, that'll sum it up a little bit better for you. So this is what's happening in glycolysis. Where is this happening? This is happening in the cytoplasm of the cytosol. And so if you go back to this diagram here, glucose oxidized to pyruvate. Along the way, we're going to reduce NAD plus to NADH, and we're going to phosphorylate ADP plus a phosphate. We're going to phosphorylate ADP to get ATP down here. This all happens out here in the cytosol. So again, to go back, the reactants of glycolysis are glucose. This is a reactant. NAD plus is a reactant, and ADP is a reactant. The final products are product of NADH, product of pyruvate, and product of ATP. This happens in the cytosol. Why? The purpose of glycolysis is to generate NADH and to generate ATP that can be used by the cell. This NADH is an intermediate energy carrier. This NADH is then going to go and be used later on in the electron transport chain. So let's talk a second there just quickly about NADH. We'd seen this in a previous screencast, right? And this is the molecule NAD plus right here. You do not need to memorize this whole thing, right? But again, this NAD plus is at a lower free energy than this NADH over here, right? So keeping in mind that like this box gets stuck onto like right here, so these two boxes, right, go together. Right? So you would have this, all of this nonsense down here would be down here. They just didn't draw that for you because that's not really the important part of this. The important part is that as I reduce NAD plus to NADH going this way, this is a higher free energy molecule. So this NADH, higher free energy molecule. Right? This is an energy carrier, and it's carrying the energy in the form of bonds. Right here, right? This bond right there. These NADHs are then going to go take this stored bond energy to electron transport oxidative phosphorylation, the later on bit there. So in glycolysis, we make these NADH molecules, these energy intermediate carriers here. This is my sort of graphic organizer mental construct. I made this, heck, all the way back when, this is my very first year of teaching this ever, like 18 years ago. This is my graphic organizer for it. So be thinking here, this is all of glycolysis, so there's two subphases, right? So what would go in this box, what would go in this box, what would go here, and what do you think is going to go in these boxes, right? And what goes there, what goes there, what goes on these arrows, so everything kind of gets labeled there, and this is the way for you to sort of practice and think about what's happening, right? So thinking over here, glycolysis means reactants, products, the location, and then an overall purpose or summary. You should print that out at a minimum and or redraw that and sort of fill those in as a way to summarize for yourself here. Nitty-gritty details, you can look at these here. There are actually 10 steps in glycolysis. You do not need to know all of these, right? But here's your energy investment phase. So here's glucose, right? We're going to use ATP to make this glucose 6-phosphate. We can use another ATP here, right? So this is the energy investment phase, and you just follow the arrow around, right? Follow the arrows around to here, right? We get this is the actual splitting right here. So this is a 6-carbon molecule. This is a 3-carbon. This is a 3-carbon, right? This G3P, right? There are 2 of them, so this 3-carbon molecule is going to be converted into this, right? So you might hear G3P. At some point, you're going to have 2 of these G3Ps after that first half. Those G3Ps then enter the second half of glycolysis. Notice that there's a prefix 2 here on everything, so everything is multiplied twice. Everything's happening twice here because you have 2 G3P molecules. So notice that all of these are 3-carbon intermediates, 3-carbon intermediate, right? 3-carbon intermediate, and ultimately you're going to end up with a 3-carbon pyruvate molecule, but you're going to end up with 2 of those pyruvate molecules there. The very last step makes a couple of ATP, so the overall net is going to be up ATP molecules. Lastly, substrate level phosphorylation. Some of you are asking about this. The substrate level phosphorylation, what's going to happen here is that there's this enzyme that's going to be a phosphorylase. Phosphorylase describes what it's going to do. It's going to phosphorylate ADP. And where does it get the phosphate from? Well, it's going to take the phosphate from this substrate that is phosphorylated. So this is a phosphorylated intermediate molecule. And a substrate level phosphorylation is that you're going to grab the phosphate from this substrate and add it to ADP, generating ATP. And then here's my dephosphorylated substrate. This will make a little bit more sense later when we talk about oxidative phosphorylation. But again, this is sort of icing on the cake if you're able to follow that there. So again, this whole screencast was dedicated to looking at this left side of the diagram there. And again, you should be able to identify the reactants, the products, the location, and the purpose of glycolysis.